Hi friends, I am Risana Fasli. Today we are discussing about xenobiotics. Xenobiotics is a chemical substance found within an organism that is not naturally produced or expected to be present within the organism. It can also cover substances that are present in much higher concentrations than are usual. Natural compounds can also become xenobiotics if they are taken up by another organism, such as the uptakes of natural human hormones by fish found downstream of sewage treatment, plant outfalls, or the chemical defenses produced by some organisms as protection against predators. The term xenobiotics, however, is a very often used in the context of pollutants such as dioxins and polychlorinated biphenyls and their effect on the biota. Because xenobiotics are understood as substance foreign to an entire biological system. Example, artificial substances which did not exist in nature before their synthesis by humans. The term xenobiotics is derived from Greek words. Xenobiotics may be grouped as carcinogens, drugs, environmental pollutants, food additives, hydrocarbons, and pesticides. Types of xenobiotics. Xenobiotics is divided into two, exogenous and endogenous. Exogenous are drugs, food additives, pollutants, insecticides, carcinogens. And endogenous are bilirubin, bile acids, steroids. Here's a picture representation of sources of xenobiotics. Xenobiotic metabolism. The body removes xenobiotics by xenobiotics metabolism. This consists of the deactivation and the excretion of xenobiotics and happens mostly in liver. Excretion routes are urine, feces, breath and sweat. Hepatic enzymes are responsible for the metabolism of xenobiotics by first activating them. Oxidation, reduction, hydrolysis or hydration of the xenobiotics are the process of that, and then conjugating the active secondary metabolite with glucuronic acid, sulfuric acid or glutathione, followed by excretion in bile or urine. An example of group of enzymes involved in xenobiotics metabolism is hepatic microsomal cytochrome P450. This enzyme that metabolize xenobiotics are very important for the pharmaceutical industry because they are responsible for the breakdown of medications. A species with this unique cytochrome P450 system is Drosophila melteri, which uses xenobiotic resistance to exploit a wider nesting range, including both soil moistened with necrotic exudates and necrotic plots themselves. Although the body is able to remove xenobiotics by reducing it to less toxic form through xenobiotic metabolism than excreting it, it is also possible for it to be converted into a more toxic form in some cases. This process is referred to as bioactivation and can result in structural and functional changes to the microbiota. Exposure to xenobiotics can disrupt the microbiome community structure either by increasing or decreasing the size of certain bacterial populations depending on the substance. Functional changes that results vary depending on the substance and can include increased expressions in genes involved in stress response and antibiotic resistance, changes in levels of metabolites produced, etc. Organisms can also involve in tolerate xenobiotics. An example is co-evaluation of the production of tetrodotoxin in the rough-skinned newt and the evolution of tetrodotoxin resistance in predator. The common garter snake in this predator-prey pair and evolutionary arms race has produced high levels of toxin in the newt and correspondingly high levels of resistance in the snake. The evolutionary response is based on the snake evolving modified forms of ion channels that is toxin act upon, so becoming resistant to effects. Another example of xenobiotic tolerance mechanism is the use of ATP binding cassette transporters, which is largely exhibited in insects Such transporters contribute to resistance by enabling the transport of toxin across the cell membrane, thus preventing accumulation of these substances within the cell. Distribution and storage. Xenobiotics are distributed through peripheral tissue via blood, lymph, cerebrospinal fluid, and then stored in liver, food contaminants, drug deposits, body fat, pesticides, organophosphate compounds, insecticides, DDT, bone, fluoride, lead, cadmium, radium, plutonium. Xenobiotics in environment. Xenobiotic substances are an issue for sewage treatment system since they are many in number and each will present its own problems as to how to remove them. Some xenobiotic substances are resistant to degradation. Xenobiotics such as polychlorinated biphenyl, polycyclic aromatic hydrocarbons, and trichloroethylene accumulate in the environment due to their recalcitrant properties and have become an environmental concern due to their toxicity and accumulation. This occurs particularly in the subsurface environment and water sources as well as in biological system, having the potential to impact human health. Some of main source of pollutions and the introduction of xenobiotics into the environment become from large industries such as pharmaceuticals, fossil fuels, pulp and paper, bleaching and agriculture. For example, there may be synthetic organochlorines and such as plastic and pesticides, or naturally occurring organic chemicals such as polyaromatic hydrocarbons and some fractions of crude oil and coal. Microorganisms may be a viable solution to the issue of environmental pollution by the production of xenobiotics, a process known as bioremediation. Microorganisms are able to adapt to xenobiotics introduced into the environment through horizontal gene transfer in order to make use of such compounds as energy sources. This process can be further altered to manipulate the metabolic pathways of microorganisms in order to degrade harmful xenobiotics under specific environmental conditions at a more desirable rate. Mechanisms of biodegradation include both genetically engineering microorganisms and isolating the naturally occurring xenobiotic degrading microbes. Research has been conducted to identify the gene responsible for the ability of microorganisms to metabolize certain xenobiotic, and it has been suggested that this research can be used in order to engineering microorganisms specifically for this purpose. Not only can current pathways be engineered to be expressed in other organisms, but the creations of novel pathways is possible to approach. Xenobiotics may be limited in the environment and difficult to access in areas such as the subsurface environment. Degradative organisms can be engineered to increase mobility in order to access these compounds, including enhanced chemotaxis. One limitation of the bioremediation process is that optimal conditions are required for proper metabolic functioning of certain microorganisms, which may be difficult to meet in an environmental settings. In some cases, a single microorganism may not be capable of performing all metabolic process required for degradation of xenobiotic compound, and so syntrophic bacterial consortia may be employed. In this case, a group of bacteria work in conjunction, resulting in dead end products from one organism being further degraded by another organism. In other cases, the products of microorganism may be inhibit the activity another, and thus a balance must be maintained. Many xenobiotics produce a variety of biological effect which is used when they are characterized using bioassay. Before they can be registered for sale in most countries, xenobiotic pesticide must be undergo extensive evaluation of risk factors such as toxicity to humans, ecotoxicity or persistence in the environment. For example, during the registration process, the herbicide Chlorantraniliprole methyl was found to degrade relatively quickly in soil. Interspecies organ transplantation. The term xenobiotics is also used to refer to organs transplanted from one species to another. For example, some researchers hope that hearts and other organs could be transplanted from pigs to humans. Many people die every year whose lives could have been saved if a critical organ had been available for transplant. Kidneys are currently the most commonly transplanted organ. Xenobiotic organ would need to be developed in such a way that they would not be rejected by the human immune system. Effects: infertility, endometriosis, dysmenorrhea, dysfunctional uterine bleeding, adenomyosis. Effects: phytoestrogens, dietary estrogen are occasionally used in natural HRT during postmenopause. Xenobiotic and human immune system. Xenobiotic may interact with the cellular immune system, which results in altered immune status, which may lead to immunotoxicity. Immunotoxicity is a picture representation, graphical representation. Characteristics of xenobiotics. Most xenobiotics are lipophilic, penetrate membrane by diffusion, transported by lipoprotein in blood, required chemical conversion to facilitate exertion. Entrance, distribution and storage of xenobiotics. Entrance to the body, ingestion, oral, food contaminants, gastrointestinal absorption, inhalation, nasal cavities, air pollutants, pulmonary absorption, skin, epidermis, dermis, toxic chemicals, dermal penetration. Other routes are injection, conjunctival instillation, suppositories. Distribution and storage. Xenobiotics are distributed through peripheral tissue via blood, lymph, cerebrospinal fluid and then stored in liver. Food contaminants, drug deposits, body fat, pesticides, organophosphate compounds, DDT, bone, fluoride, lead, cadmium, radium, plutonium. Excretion of xenobiotics. Urinary excretion, biliary excretion, exhalation, other secretions, saliva, sweat, breast milk. Thank you.